Good afternoon, everyone. Please wait just a minute.
Good afternoon.
I try to open my presentation. Okay, thank you for your waiting, and let's start our lecture number 6. The title is Selection of Average Samples of Various Types of Raw Materials and of Food Products. And the purpose of this lesson is to familiarize students with the selection of average samples of various types of raw materials and finished products. And then next, about the plan of this lesson. Today, I will talk about the average tests, about the dumplings, and how to take the average samples from various types of raw materials or food products, for example, from eggs, from fish, and the honey, milk, and the meats, and the other products. And the first introduction to the lesson. In order to proceed to the study of any raw material or product, it is necessary to correctly select samples for examination. Yes? And in the processes of quality control of food products, a small part of them is spent. And the amount spent on testing the product depends on the size of the batch and the parameters of the study of these products. And there are difference between organoleptic or other name is sensory and the laboratory methods. And the difference is that when determining the appearance, conditions of the container, and the packaging size, for example, and the shape of this product, and some organoleptic parameters, test samples is not consumed. And the average sample selected from the controlled batch of food products should reflect the overall conditions of the entire batch. If these conditions is not met, the most ideal studies will be erroneous. And for analysis, select a part of product from homogeneous batch from the product developed at one enterprise in one shift by one team, and or not products of the same name from the same enterprise received on one invoice. And all this determined by an external inspection of the party. And after establishing the uniformity of the batch, the date of the accompanying documents are compared with the inscription of the label on making, on the container, and etc. And sometimes it's the result of non-compliance with the rules of transportation. The batch becomes heterogeneous. For example, product are weak, and etc. And such a batch is stored in the 2 or more days. And the sampling techniques depends on the functional state of the test product. For example, the raw materials or food products may be solid conditions, bulk, liquid, maybe other conditions, and the average sample is taken together with the supplier or with the participation of a representative of the quality inspectorate or with a representative of local authorities. And for examination at a meat processing plant with a conditions process of meat processing, workplaces for veterinarians should be equipped to inspect internal organs, carcasses, and lymphatic submandibular nodes. And without veterinarians, livestock processing is not allowed. Next slide about the how to determine the meat or meat products. Suspicious carcasses are separated and examined for infectious disease. Yes, and for example, for organoleptic biochemical studies, samples weighing at least about 200 grams are taken in a single piece from each suspicious carcass. And the sample should contain bone, marrow with bone, tendons, and the fat. And the for chemical studies each sample is separately brought 3 times in a meat grinder with 3 or 4 millimeters stir and thoroughly mixed. And to grind the meat using the meat grinder, mixer, homogenizers, mortar, and other apparatus. And the samples taken for research in moisture-proof container and sealed with an accompanying documents sent to the laboratory. Next, about the fish for laboratory tests are selected from different places, at least 5% of the lot of caught fish. Boxes, bags with a weight, of up to 100 grams from 5-7 pieces from the batch of package, and up to 1 kilo, 1 or 2 kilo, 2 samples of 100 grams from 2 fish from each batch, and up to 3 kilo from the 2 samples of 150 grams from 1-2 fish from each batch. And the next slide about the sausage. Sausage samples from the sausage products are taken from each homogeneous batch. And the cut of samples with a length of 15 centimeters each from the edge for the loaf, from the hams along the entire thickness with a width of 10 centimeters from the tongues to units of production, and the samples are crushed and examined. And the samples taken for research in moisture-proof container and sealed within a accompanying document are sent to the laboratory. And the dumplings is in the amount of 1% from 1, but not 1,000 grams. And the sampling the takes of sampling from sausage according to the cost 97 to 97, 92, and 73 sausage products and the products of pork, mutton, beef, and the meat or other kinds of slaughtered animals and poultry. And acceptance rules are the same methods. This is the name of this cost. And from selected units, single samples are cut off in the transfer direction at a distance of at least 5 centimeters from the edge. And the offsets, there are general samples, one for organoleptic, the other for laboratory tests. And the sample weight for organoleptic studies of sausage quality is 4 or 500 grams, and for chemical studies, it must be about 2 or 250 grams. Next slide about the canned products. For the study of canned food, select 2 units of packaging for organoleptic evolution and 4 for chemical studies with a container capacity of up to 1 liter and over 1 liter to select 2 units, and above 3 liter are carried out in 3-liter containers after taking a sample for bacteriological analysis. And for the studying the canned products using the GOST number 34177 2017, and the name is canned meat general specifications. Next, in the next table, you can see the sampling of fresh and freshly frozen fruits, berries, vegetables, and the table greens. For example, from the table greens, it must be various types, yes. The table greens may be dill, parsley, coriander, green onions, celery, chives, and etc. And from these products, we can take the 0.1 kilos for the studying. And from the leafy vegetables, from the, for example, salads, spinach, cabbage salads, various herbs used in food, we take 0.1 kilos too. And from mushrooms, 0.1 kilos too. And from vegetables, for example, from cabbage, lettuce, and etc., we must take with 10 to 15 copies upper and basal leaves. And from the cucumber, tomatoes, sweet peppers, and from the potatoes, beets, carrot, onions, we take 0.5 kilos. for the analysis. Okay, and the from next product is melons. We take is washing off the surface of 20, 25 copies. And from fruit and the berry products, for example, from apple, pears, cherries, plums, avocados, citrus fruits, and the other types of fruits, we can take 0.5 kilos. And from nuts, without sugar, we take 0.1 kilo or 2 units of packaging. And the next slide about the eggs. And for the taking, you use gold standards 30363 2013. The title is Egg Products, Liquid and the Dry Food and the Technical Conditions. And the frozen egg products selected uses special proof from 6 cans in the 4 placed in several corners to the bottom. The weight of the average sample is not more than 1 kilo, and they are served at the temperature of 15 degrees and mixed. And the egg powder samples are taken with a probe from 10% of units. And the packaging and the average samples weighting must be 250 grams. Next, about the taking the samples from drinking water. In our country, use the cost of Russian Federation. The number is 56237 2014. This title is Drinking Water, Sampling on Water Treatment Stations and in pipette distribution systems. And the water is tested for at least 2 hours after sampling, transported at the temperature of 5 degrees. In exceptional cases, no more than 6 hours. And the samples are taken in a vial with a lipid stopper with a capacity of up to 0.5 liters. And for studies on the presence of pathogenic microflora up to 1 liter. And the water from the tap is lowered within 10-15 minutes. And there are 6 rules which you can use when you're taking the samples from water for analysis. For example, rule number 1. When taking a sample of water for chemical analysis, use a clean bottle, plastic or glass, with a volume of 1.5 liters from drinking or mineral water. And you must never use bottles of sweet flavored beverages. Okay, understand? Like this picture, you can use this like these bottles. And rule number 2. Before collecting water, it must first be dried for 5-10 minutes. If the water hasn't been used for a long time, it may take longer to drain the water. This must be done in order to avoid getting the stagnant water in the samples. Next rule, before sampling, the bottle and the stopper are thoroughly rinsed several times from the inside with the water that will be taken for analysis. In this case, you cannot use detergents like this picture. Okay? Next, rule number 4. It is desirable to collect water in a thin stream and along the wall of the bottle. And this method of selection reduces the saturation of water with oxygen in the air and prevents chemical reactions. Rule number 5. The selected water sample should be delivered to the laboratory as soon as possible, and the faster the water is received for analysis, and the more reliable the results will be. And the next, the rule number 6 is if necessary, provide the water sample with a accompanying document indicating the place of selection. For example, it may be city, village, street, house, and the next indicating is water source, maybe well, spring, artesian from a well, and the water supply. Time and the date of selection, maybe date, months, and the water packaged and in a container. If possible, is delivered to the laboratory in the factory, undisturbed packaging, and if this problematic, then bottled water is poured into a plastic container from under distilled water. And there are only 6 rules which you may using when you take the samples. And the next presentation is about the honey. Honey, if there are several containers, samples are taken from each. The liquid honey is first mixed. The average sample is taken with a tubular aluminum sample with a diameter of 10, 20, 20 millimeters and some verging it over the entire length of the container. The rules of veterinary and the sanitary examination provide for selecting 100 grams of honey from each controlled unit of packaging. And for determining the water content, about the 200 grams after organoleptic laboratory tests, and the remains of samples are not returned to the owner and they are sent for technical disposal. And then using the cost of Russian Federation 54644 2011 and the name is Natural Honey Technical Conditions. And according to this lesson, you can use many words, many new words. For example, dumpling. Yes, dumpling is a broad classification for dish that consists of piece of dough made from the variety of starch source and wrapped around the filling or dough with no filling. And the dough can be based on bread, flour, or potatoes, and may be filled with meat, fish, cheese, vegetable, fruits, or sweets. And the dumplings may be prepared using a variety of methods, including baking, boiling, frying, simmering, steaming, and found is many words cuisines. And egg, for example, egg is organic vessel containing the zygote in which embryo develops until it can survive on its own, and at which point the animal hatches and an egg results from the fertilization of an egg cell. And the most arthropods and the vertebrates, excluding live breeding mammals and the mollusks, lay eggs. Also, some such as the scorpions do not. And the next is fish. Fish is breeding aquatic uh, craniate animals that lack limbs with digits, and they form a sister group to the tunicates and together forming the olfactories and includes in the definition as a living hagfish, lampreys, and the cartigenous and the bony fish as well as various extinct related groups. And around 99% of living fish species are ray finned fish belonging to the class Actino, Actinopterygii, and with over 95% belonging to the Telos subgrouping. And the next is honey. It's a sweet, viscose food substance made by honeybees and some related insects. Bees produce honey from the sugary secretion of plants or from secretions of other insects by regurgitation, enzymatic activity, and water evaporation. And the bees store honey in wax structures called honeycombs, and the variant of honey produced by honeybees is the best known due to its worldwide commercial production and human consumption. Honey is collected from wild bee colonies or from hives of domesticated bees and the practice known as beekeeping or apiculture. And the next is milk. Milk is a nutrient-rich liquid food produced by the mammary glands of mammals, and it is a primary source of nutrition for young mammals, including breastfed human infants before they are able to digest solid food. And thank you for your attention. Our lecture is over. If you have questions, please ask. Сұрақтарыңыз болса, қойсаңыздар болады. Do you have any questions?
No.
Okay. Так, біті 18-11 штук қана. Біті...
Я бармыз апай.
Так, Рашимов Тайгер сабаққа кірді ме? Староста?
Жоқ, кірмеді.
Кірген жоқ. Так, қалған студенттерді жазып жібересіздер ме?
Аха, жүреді апай.
Біті 18-12.
Ала апай, саламатсыз ба?
Саламатсыз ба? Так, Жанбауып Медет, Күлбарак Нұрдана, Елнұр, Жандос, Марлен сабаққа бармадың?
Бармын апай, Нұрдан.
Нұрдана бар. Ұлдардан ше? Ұлбалладың. Так, өте 18-16. 5-ші аптада болған жоқ Құттымұрат Айдана.
Ұлдардан.
Ниязбекова Анель және Ермахан Берікұлы. Бүгін бар ма сабақта?
Елнұр бар.
Апай, Құттымұратова Айдана бар сабақта 18-16. Апай, бұты 18-12-ден Ижанбауып Медет, Темірхан Жандос, Адвокатов Елнұр, Тұрсынбаев Марлен, Екүлбарақ Нұрдана бәрі бар. Бәрі отыр.
Түгел 26 студент түгел ма? 12?
Я тексеріп қарадым, бәрі түгелінен отыр.
Маған скриншотын беріңіздер, е?
Хорошо.
18-21. Пускай группа. Так, Утигенова Ахем. Есть? Так, Ахмад, староста. Утигеновой нету, да? Шизяцин. Гейн. Так, жарайды. Маған скриншот жіберіңіздер, старосталар. Қатысқандарыңызға рахмет. Thank you for your participant and you may go out from this conference. Goodbye.
Thank you.
Goodbye, teacher.
Goodbye.